Welcome back to the Deep Dive. Today, we're going to dig into some really remarkable science, stuff that might just explain a bit more about what makes us, well, uniquely human. Imagine scientists piecing together this ancient puzzle, this kind of riddle hidden right there in our DNA. Our deep dive today focuses on some cutting-edge findings. We're drawing from a piece called Human Evolution, Brain Chemistry and Competitive Advantage, and also a detailed research article from PNAS. And together, these sources trace these really subtle but potentially profound changes in brain chemistry across, well, the whole human family tree.
Exactly. And our mission today really is to unpack the insights, the subtle but maybe incredibly significant things hidden in these sources. We're zeroing in on how these tiny biochemical shifts might have played a role in our species' success, especially after our lineage split off from Neanderthals and Denisovans maybe 500,000 years ago, give or take.
Yeah.
So we'll look at what these changes actually are, what effects scientists have seen, at least in experimental models, and what big questions they bring up about our past, and maybe even our present.
It's a fascinating area. So where do we start with this biochemical mystery?
Well, it seems to kick off with an enzyme called adenylosuccinate lyase, or ADSL for short.
ADSL. Okay, sounds complex.
Yeah, it does sound a bit technical, but it's crucial for something absolutely fundamental, making purines.
Purines, right. Those are the building blocks for DNA and RNA.
Exactly. DNA, RNA, other vital bits and pieces that, keep everything running.
Okay, so we're really core biological process.
And what's really interesting here is that researchers from OIST in Japan and the Max Planck Institute in Germany found that really tiny changes to this specific enzyme, ADSL, might play a pretty significant role in our behavior, and maybe even in what defines us as modern humans.
Wow. So what kind of changes are we talking about? You said tiny.
Incredibly tiny, actually. That's what's so remarkable. So the ADSL enzyme, it's this chain, right? 484 amino acids long. Okay.
The modern human version differs from the ancestral one, the one we see in Neanderthal and Denisovan genomes, by just one single amino acid.
One out of 484.
Just one. At position 429, the ancestral form had alanine. Modern humans have valine there instead.
That's it, that tiny switch.
That seems to be the first key change. And what's striking is that this valine version, it's present in virtually all present-day humans.
Huh. Which tells you it must have spread pretty quickly after we diverged from Neanderthals.
Exactly. It looks like it appeared after that split, but before modern humans migrated out of Africa. So it was an early foundational change for our specific lineage.
Okay, so one tiny amino acid swap. What did it actually do, functionally speaking?
Well, in the lab, when they tested it in vitro, this single substitution makes the ADSL protein a bit less stable.
Less stable. Okay.
And when they introduced this human variant into mouse models, it led to higher concentrations of the stuff ADSL is supposed to process, its substrates.
It's a backlog. Sort of, yeah. A buildup of its raw materials, particularly molecules called SACAR and SADO, and this buildup was seen in several organs, but it was especially noticeable in the brain.
Why the brain specifically?
Well, it seems ADSL expression, the amount of the enzyme produced, is naturally lower in the brain and also the liver compared to other tissues. So these organs are probably more sensitive if the enzyme's activity dips even slightly.
Okay, that makes sense. So a less stable enzyme leads to a buildup of its ingredients, especially in the brain. Did this actually change how the mice behaved?
It seems so, yeah. They did some pretty interesting behavioral experiments. They used something called an IntelliCage system.
IntelliCage? Sounds fancy.
It's basically a sophisticated setup, yeah.
Yeah.
Allows researchers to monitor complex mouse behavior in a controlled, sort of semi-naturalistic group setting. And what they did was restrict access to water.
Creating competition.
Exactly. Making water a scarce, contested resource. And what they found was that the female mice with the modern human ADSL substitution, well, they consistently got to the water more often and more efficiently than their wild-type litter mates.
Really? How much more efficiently?
The paper mentions, for example, during a 3-hour window when water was available, these humanized females made about 1.8 times more visits, at least for the first few visits, compared to the controls.
Wow, that's a noticeable difference in a competitive situation.
It is, and this difference held up even when they made it tougher, like restricting water to just one specific corner of the cage. The humanized females still showed this competitive edge in getting to it.
But you specifically said female mice.
Yes, and that's one of the really intriguing parts. They didn't observe the same competitive advantage in the male mice.
Any idea why?
The sources suggest one possibility. It seems that the female humanized mice actually expressed less ADSL protein in their muscle tissue, about 40% less compared to the humanized males.
So maybe the females were even more sensitive to the effects of this less stable enzyme.
That's the idea. A heightened sensitivity in females might explain why the behavioral effect was prominent just in them, at least in this specific test.
Interesting. And they ruled out other factors, like were these mice just generally stronger or more dominant?
Good question. They checked that. The humanized mice didn't show any inherent dominance in standard tests, like the tube test, nor did they show improved muscle strength overall. So it really points towards a specific competitive advantage related to, well, resource acquisition strategy or efficiency rather than just being bigger or tougher.
Fascinating stuff in mice. But okay, here's the million-dollar question, right? What on earth could this mean for us, for humans? These are mouse studies, after all.
Absolutely crucial point. You can't just map mouse behavior directly onto humans, but the story doesn't actually stop there. There's another layer to this ADSL evolution.
Oh, what else did they find?
Well, the researchers identified a second set of genetic changes affecting ADSL, but these weren't in the main coding sequence, the part that spells out the amino acids.
Where were they then?
They were in a non-coding region of the ADSL gene. Think of it like the dimmer switch or the volume control for the gene. It doesn't change the enzyme itself, but it affects how much of it gets made.
Okay, regulatory changes.
Exactly. And this set of variants found in this regulatory region is also incredibly common today, found in at least 97% of present-day human genomes.
97%. Wow. So another widespread change. Did this one maybe compensate for the first change? Did it boost ADSL activity back up?
You'd think that might happen, right? Evolution trying to balance things out. But no, surprisingly, these non-coding variants seem to do the opposite.
They reduce activity even more.
That's what the evidence suggests. They appear to further diminish ADSL activity, mainly by reducing the amount of ADSL RNA that gets produced. And again, this effect seemed most pronounced in the brain.
So wait, we had one change making the enzyme less stable, reducing activity. And then another set of changes, also widespread, that reduced its production, further lowering activity.
Precisely. As the researchers put it, the enzyme seems to have undergone 2 separate rounds of selection that reduced its activity.
2 rounds, both pushing activity downwards.
Mm-hmm. And importantly, both the original amino acid substitution and these later non-coding changes show strong statistical signals of positive selection.
Meaning evolution actively favored these changes. They must have provided some kind of advantage to early modern humans.
That's the strong implication, yes. An evolutionary advantage significant enough for these changes to sweep through the population.
Okay, let's try and bring this back to humans again. We have these 2 sets of changes, both selected for both reducing ADSL activity, especially in the brain. But isn't having too little ADSL bad? Don't genetic ADSL deficiencies cause serious health problems?
Yes, absolutely. That's a critical piece of the puzzle. Complete or near-complete loss of ADSL function due to severe mutations is known to cause a devastating disorder.
What kind of disorder?
It leads to severe developmental problems, psychomotor retardation, often aggressive behavior, features of autism, intellectual disability, and really significant speech and language impairments.
Okay, so clearly having 0 or very low ADSL activity is detrimental.
Extremely detrimental, which highlights this really delicate balance, doesn't it? The modern human variants, the ones that were selected for, they reduce ADSL activity compared to the ancestral form.
Right.
But they keep it active enough to avoid those severe pathological deficiency disorders. It suggests there might be a kind of sweet spot, an optimal range for this enzyme's function in humans.
Tuned down but not off.
Exactly. Tuned down just enough, perhaps, to confer some benefit without causing disease. And there's another piece of human data that fits here.
Look at that.
The researchers looked at levels of SA-DA, remember? That's one of the substrates that builds up when ADSL activity is lower.
Yeah, the backlog molecule.
Right. They looked at SA-DA levels in human cerebrospinal fluid, the fluid surrounding the brain and spinal cord, and they found a significant negative genetic correlation between higher SA-DA levels and scores on intelligence tests.
Whoa. So higher SA-DA, which implies lower ADSL activity, correlated with lower intelligence scores.
Yes. The correlation wasn't huge, Rg equals 0.13, but it was statistically significant. Now, correlation is not causation. We have to stress that.
Of course.
But it does provide a hint, a piece of human-specific data suggesting that variations in ADSL activity might indeed affect cognitive functions in humans, potentially linking back to what was seen with the competitive behavior in mice, maybe? It's speculative, but intriguing.
It is intriguing, but it's so important to add the caveats, isn't it? Mouse findings don't just map onto humans one-to-one.
Absolutely essential. Human behavior is incredibly complex. It's shaped by countless genes, environmental factors, culture, you name it. And the precise mechanisms linking ADSL activity to behavior, whether competitive or cognitive, are still largely unknown. We're really just scratching the surface. The specific effects would be embedded in this whole complex web.
So lots of open questions still.
Definitely. These findings really open up more questions than they answer in many ways, like why only the female mice showed that competitive advantage in that specific water task. That remains unclear.
Yeah, that's a puzzle.
But there's a broader takeaway here for the science itself, highlighted in the sources, which is that to really understand complex human traits, especially using experimental systems like mice, scientists might need to model multiple genetic changes that affect a particular pathway or system, not just tweak one gene in isolation, but perhaps recreate combinations of changes, like these two hits to ADSL activity.
Ah, I see. To capture the cumulative evolutionary effect.
Potentially, yes. It might give a more realistic picture.
So if we kind of zoom out, the big picture here is that we're starting to piece together how human metabolism, our basic biochemistry, has actually changed over the last 500,000 years or so.
That's right.
And this enzyme, ADSL, it's one example, maybe one of a small but important group of enzymes that evolution has tinkered with in our lineage.
It's a fascinating window into our biochemical evolution.
Wow, what a deep dive that was, really getting into the nitty-gritty of our human blueprint. We went from a single amino acid swap
So one tiny change.
to then these later non-coding genetic tweaks.
Yeah.
And seeing how both sets of changes, apparently favored by positive selection, work together to gradually lower ADSL enzyme activity in modern humans compared to our ancestors.
And the implications are pretty profound, aren't they? It's not just some obscure detail about purine biosynthesis. It's about these subtle biochemical shifts that maybe, just maybe, influence things like our competitive behaviors, perhaps even cognitive abilities, things that might have helped distinguish our lineage from our close ancient relatives like Neanderthals and Denisovans. It kind of challenges the idea that you always need huge genetic revolutions for significant evolutionary change.
Yeah, it really does. It makes you wonder, doesn't it? Well, what if the chemistry that really makes us human, isn't about some massive sweeping genetic overhaul? What if it's more like a series of these incredibly precise, tiny adjustments, like fine-tuning a complex machine, each little nudge subtly pushing us towards the unique creatures we are today?
It's a compelling thought.
And it leaves you thinking. What other hidden biochemical stories like this one might be encoded in our genes, just waiting for scientists to uncover them?